Today's lecture is entitled Structure Elucidation of Compounds Isolated from Plants. The learning objectives are the following. Steps involved in the isolation of phytochemicals, an NMR spectral analysis of chemical isolates to determine their structure. At the end of this lecture, you should be able to identify the steps in the process of isolation of phytochemicals, explain the process involved in each step, state how the isolated phytochemical is processed using NMR spectroscopy, and give an example of how this was done for a known phytochemical. The picture on this slide shows a man looking at a haystack with a needle in it. And it's important because oftentimes when we are looking for phytochemicals in plants, it is likened to looking for a needle in a haystack. The isolation of phytochemicals entails a number of steps. These steps include the following. Starting with identification of the plant part to be studied, a harvesting phase is undertaken. Now, this harvesting phase entails accessing of the plant part in bulk quantities. And it is followed by a drying step, a maceration step, an extraction step, and finally an isolation/analysis step. The harvesting step entails, like I stated previously, the accessing of the plant part in bulk quantities. Pictured here in the upper left corner is that of a tree that is the source of the plant part. And in this particular case, the plant part of interest is the leaves. Once the leaves are isolated in bulk, they are harvested and gathered in large gallon-sized bags, following which they are subjected to a dehydration step where they're laid out on a flat surface to allow for removal of any water that's present within the leaves themselves. After a predetermined amount of time, the dried leaves are then gathered together and then weighed. In this particular case, the plant part would have actually amounted to about 2 kilograms after drying. Once dried, the leaves are subject to a maceration process. Maceration entails the pulverizing or grinding of the plant part so as to increase the surface area, which would then facilitate optimal extraction or removal of the phytochemicals that are present within that plant part. Once the maceration step is completed, the plant part itself that has been pulverized is then transferred to a large glass jar and applied with solvent, polar solvent, that is utilized as a mobile phase to remove all of the phytochemical components of that plant. This is done over a period of about 24 to 48 hours and is referred to as a slow extraction process that is intentionally left at room temperature so as to minimize degradation of the phytochemicals. Upon completion of the extraction process, the solvent, as you can see, would have become discolored as it takes on the colors of the actual plant extracts or the phytochemicals that are now being removed from the actual plant part residue and now entering into the mobile phase, which is the solvent. At this point, since that extraction process is completed, it is now possible to separate the solvent or the liquid phase itself from the initial plant part that was placed there to be extracted. And that separation process is referred to as filtration. And it is a physical separation that takes place by way of a filter funnel and a flask. So that is pictured in the upper right corner, where you can see the plant residue is actually isolated and collected in the funnel itself, while the solvent that contains the colored phytochemicals are filtered into the bottom of the flask. The component of interest, of course, is the solvent itself, as it actually would have been stated before, is the vehicle or the mobile medium by which the phytochemicals themselves have been removed. Once we separate the solvent from the plant residue, which is in the funnel at the top, we're now able to conduct a further separation. But this time, that separation is by way of heat. So at this point, the lower picture in the lower right corner is depicting the removal of the solvent, because at this point the solvent is no longer necessary since it has done its job as a vehicle to remove the actual phytochemicals from the original plant part itself. Having done this, we can now remove the solvent by evaporation. And having done that, a further step is now allowed where we can get to our residual plant isolate. In this particular case, we would have started with 2 kilograms of plant parts. And by the time we've carried it through the series of steps, we find that we're at a place of 8 grams in terms of mass. Once the 8 grams have been obtained, it is further processed through another series or sequence of steps, 2 of which are referred to as chromatography. The word chromatography actually is defined as separation by colors. And in this case, what we're actually doing is we're separating the plant isolate on a more molecular or chemical level, because at this point you can see that the phytochemicals that are present have been physically removed from the plant parts. All the cellulose material and all those fibrous components of the plant itself, those are no longer present. And at this point, the residue or the plant isolate is primarily composed of phytochemicals. So these phytochemicals are there in bulk, and they now need to be separated from each other. In order to do this, we have to conduct 2 chromatographies. The first one is pictured on the upper right corner of the slide, and it is referred to as thin layer chromatography. In this particular chromatography, there isn't any removal of components. This chromatography is simply conducted to check to see what the relative separation behaviors are for each phytochemical. And that's depicted on the thin layer plates that are present in the beakers at the top. Once we get a picture of what those separation behaviors are like, we are now in a position to physically separate the components, and that is conducted by way of a column chromatography. And so you can see the lower right-hand picture shows a glass cylinder, which is referred to as a column, that contains a white component, and that is actually the separation medium. And at the bottom of that glass column, there are some collecting flasks, and those are actually the containers that are used to collect the individual phytochemicals as they are being separated from each other. Eventually, that solvent is removed, and we get to the picture that's on the lower left corner, which is actually the dried isolate of pure phytochemicals. Now, I have a picture here on this slide which gives you an indication of the summary of steps, but this is by no means as simple as it looks. Many times we have to repeat these steps multiple times over. Following the actual isolation of the pure compound, we're now in a position to prepare the sample for spectral generation. What that means is that we would take the sample itself, because we've isolated a pure compound, but we do not know exactly what it looks like in terms of its chemical composition. In other words, how do the bonds connect to each other, and where are they positioned? That information is obtained by way of a spectrometer. And so in order to be able to use a spectrometer, we have to prepare our compound in such a way where it can be analyzed appropriately by the instrument. And so depicted in the upper right corner is a picture of that sample in the corresponding medium needed for the instrument to be able to read it properly. So the picture specifically actually shows what is called an NMR tube, and that's actually a capillary tube that is then designed specifically for the instrument. The lower right-hand corner shows the actual NMR instrument itself, which in case is a magnet, that is a super magnet that is used to then distinguish the electronic environment of the various components of the molecule. At the end of the analysis, the spectrum is generated, which is pictured on the lower left corner, and it actually is a spread of the proton atoms on the actual molecule itself according to their varying electronic environments. It's very specific and therefore very detailed. And based on the spread, we are able to determine what kinds of protons we have and what ratios and relative positions they exist in. So, having generated our spectrum, we're now in a position to be able to connect the spectrum to, as in spectral peaks, to the actual compound structure itself. And so we see that depicted here in a color-coded fashion, where the purple peak, or the peak that is circled with the purple circle, is actually connected to the proton that is seen as He in the molecule. And then we see the peak that is encircled red is connected to the protons that are circled as red in the actual structure, and the same goes for the peaks that are encircled in blue. So this is how we correlate the spectrum to the actual structure of the chemical itself. And of course, there are other instruments that we engage to help us to corroborate this information. But specifically today, we're talking about how NMR spectroscopy connects with structure. So at the end of this lecture now, it is our hope that you've been able to identify the steps in the process of isolation of phytochemicals, explain the process involved in each step, state how the isolated phytochemical is processed using NMR spectroscopy, and give an example of how this was done for a known phytochemical. This concludes our lecture today.